Hi, I'm Meredith. I'm a dietitian. I'm probably not your dietitian, so please consult with your own personal health care provider before you make any changes to your diet, your supplements, your medications. This video is to explain why I think the anhydroretinol is a cause of a lot of the symptoms of vitamin A toxicity. I'm not exonerating other accumulations of forms of vitamin A that could potentially be causing damage to the body. I have a whole map called the retinol aldehyde map, and I was gonna release it, but maybe it was fate that I didn't, although all of the things in there still apply. So that's pretty interesting. I just need to kind of make it a little bit more user friendly because it's massive and it's like impossible to print. So, so yeah. So when I found out about anhydroretinol, I was like, is this it? Is this the problem? So I decided to kind of go through what I think would happen in the mitochondria if there was anhydroretinol in there. So let's do this. And yeah, I'm gonna get giddy, but it's my way of dealing with sadness. So I'm one of those people that laughs when someone breaks their leg. It's horrible. So if you see me tittering, it's not because I don't think this is bad, it's because I'm a weirdo. Okay, so this is the paper from Kim at all those guys. Actually, there's two authors here, Kim and Hammerling. It's called the Mitochondrial PKC delta retinol signal complex exerts real-time control on energy homeostasis. And this is a 2020 article. And a shout out to Theo, because I would not have known that this existed if she hadn't sent me a podcast to listen to. And I don't usually listen to very many podcasts. Not that I wouldn't, I just don't really have time for it. So I listened, and the people mentioned this paper and the fact that you needed to have cod liver oil because you need retinol to be able to work your metabolism correctly for oxidative phosphorylation. And I was like, wow, that is so cool. So I don't get stuck in paradigms, by the way. Like if I mess up on something, I'm like 100% like, okay, yeah, I didn't know that. Like I can't know everything. I wish I could. Gosh, I wish I could. So I used to think that retinol had no role in the body, but it does. And this paper proves it. So, okay. So retinol takes a special place in the electron transport chain as a signalome. And so what it does is it senses how many electrons are in the intermembrane space of the mitochondria. And if there are too many electrons, then it tells the enzyme, in a roundabout way, it tells the enzyme pyruvate dehydrogenase complex to turn off. There's plenty of electrons in the cell. We don't need any more. We're going to make plenty of energy. Stop pushing pyruvate through to acetyl-CoA. But if there are not enough electrons, then this signalosome would actually tell, in a downward cascade, tell pyruvate dehydrogenase complex to turn back on, so that there would be more pyruvate going into to acetyl-CoA. So you'll see that in just a bit. So there's a reason why it has to be retinol, and that has to do with another study by the same group that's quantum mechanics. I'm not a quantum mechanics expert, so I'm just kind of going along with what the paper says. And so if you are an expert in quantum mechanics, I'll leave a link in the show, in the description, and you can check out this paper, both of these papers. Okay. So let's real quick talk again about the different types of vitamin A that we're dealing with in the signalosome. So we have retinol, and retinol is what the body prefers to put in the signalosome. And the quantum mechanics study says that it requires specific double bonds to work properly. So if you look at retinol, there's a double bond in the beta iodine ring, but I don't think that's the bond that they're concerned about. I think it's more of these bonds that are along this sort of tail. And you can see that I mean, you're going to have to look at the video if you're listening. The orientation is a specific way for retinol. And also retinol has a hydroxyl group here at the end. That's important because you can't go from, you can't use alcohol dehydrogenase to convert retinol into retinol aldehyde if it doesn't have a hydroxyl group. So let's look at anhydroretinol. We can already see that there's a shift in the double bonds. And so the authors of the quantum mechanics paper say this is the cause of anhydroretinol not working well because the electrons flow over the double bonds in the signalosome. And if the double bonds have shifted, there's a problem. There's also a lack of a hydroxyl group. So anhydroretinol is not a functional form of vitamin A. It does not have a role in the body. It is pathological. Okay, so rehydroretinol is a version of vitamin A that we can make from anhydroretinol. The one study that I have read, I've read a couple studies about this, but they used vitamin A deficient rats. So the rats wanted to convert anhydroretinol into rehydroretinol. And this occurred in the absence of alcohol. So if we think about it, retinol is a primary alcohol. And so I think that it's potentially an issue that if we have very, very high retinol levels and it's not bound, then retinol can serve as its own primary alcohol. And it's the presence of alcohol, I believe, is the cause of the making of anhydroretinol. So that'll be a future video on how to kind of be better at controlling the amount of alcohol available to kind of catalyze the conversion of anhydroretinol into rehydroretinol. So you need an alcohol and an acid. We have acid all the time in our body, but we shouldn't be having alcohol in our body all the time. But you can see that rehydroretinol, again, the double bonds have moved. And so it's likely not effective like retinol. And some of the studies say that it's like has 7% of the activity of retinol. So we want to prevent the formation of anhydroretinol. And we definitely don't want anhydroretinol in our PKC delta signalome. And then another study shows that we can convert from anhydroretinol into 14-hydroxy-4-14 retroretinol. So it's kind of like good versus evil. So this one actually can cause growth in certain cell lines. Obviously, anhydroretinol has been implicated in killing cells. We can actually use it for cancer to kill a cancer cell, but it will also kill a regular cell. So I think of it as evil anhydroretinol versus good 14-hydroxy-4, 14 retroretinol versus excellent retinol. And then rehydroretinol is meh. So it's just a meh. It's not a very good compound. Okay. So this is a cell, and here's the cytosol. That's the outside. And then in the cytosol, we have a reaction called glycolysis. We go from, I didn't write it on here, but we go from from glucose to pyruvate. And then within our mitochondria, we convert pyruvate to acetyl-CoA. And acetyl-CoA is where our fat metabolism comes in. So when we're using fat as energy, it enters here at acetyl-CoA. It doesn't go through pyruvate dehydrogenase complex. So we have some regulation components to this. So again, when the cell levels of electrons get high in the intermembrane space, then the signalosome senses that, and it will lead to a cascade that turns off pyruvate dehydrogenase complex. And then when the electron levels go down in the intermembrane space, it will lead to a cascade of events that turns on pyruvate dehydrogenase complex. Okay, so once you go through, you have acetyl-CoA, and then you go through the TCA cycle, you make some NADH, and then at the same time that you make NADH, you also have some electrons that are building up, right? And then the electrons are they're passed along here, and I added in some more detail in another slide. So let's look at what happens for the articles when you have the wrong type of vitamin A, right? Okay, so here is PKCδ. And it's got retinol in it. And the researchers in this particular paper, which is Quantum Chemistry Rules Retinoid Biology. In this paper, in this study, they show that when you have retinol in the PKCδ, it becomes both active and inactive. It does the correct type of sensing to control oxidative phosphorylation, or the making of ATP. But when you have anhydroretinol in the PKCδ signalosome, instead of it having an inactive component, it just stays active and it stays on. And this can cause massive damage to a cell. Okay, and we'll see that. And then if you happen to have anhydroretinol mixed with a bit of retinol in the PKCδ, it just serves as, it goes into an on phase, but it's extended. So it's sort of like an energy boost for the cell. And so the authors think that that might be the reason why it's okay to have a small amount of anhydroretinol. It'll be like a little energy boost, right? And then we go back to normal. So they also implicated apocarotenols in this, especially the beta apo-10 carotenol, and that's made by BCO2, an enzyme that's in the mitochondria. If you have a slow BCO1, that can make you at risk for making extra of that beta apo-10 carotenol. And then there's another one, beta apo-13 carotenone. It hasn't been implicated in this pathway, but it can mess with your ability to get rid of retinoic acid. So that could lead to a buildup, I believe, like a feedback and cause some issues. So, again, I'll say this. If you have retinol around to buffer these two, nasty forms of, or nastier forms of vitamin A, if they're in excess, then you're doing okay. It's when you start to have, and what I think is when you start to have too much of anhydroretinol or the apocarotenol, and that's keeping you from being able to have normal metabolism. That's the problem. Okay, so now let's look at this a little bit closer. Again, we have pyruvate that comes from a glycolysis, and it goes through pyruvate dehydrogenase complex. Excuse me. And then you move on to, you make acetyl-CoA, and acetyl-CoA goes through the TCA cycle. You produce NADH, and then that's recycled at complex 1 to NAD, and it actually pushes a hydrogen over here into the inner membrane space. The electrons are transferred along coenzyme Q10, and they finally end up at complex 3, and then complex 3 transfers the electrons to cytochrome C, and then cytochrome C transfers the electrons to complex 4, where you make water and half of a O2 molecule. And then the end process is the hydrogen ions that have built up through these different complexes. They get pushed through ATP synthase, and you make ATP. So the goal, the role of the signalosome is to sense the electrons, because knowing how many electrons are in the inner membrane space will tell, whether or not you should keep this energy cycle going. And we're going to see that if you can't stop the energy cycle, there's a problem. So with adequate retinol, anhydroretinol will serve as just a temporary energy boost to a cell, but when there is a lack of retinol to buffer, then there's pathological activation. Okay, so I made little notes on the screen so that I don't get off track, because you probably can tell by my videos that I get super excited and I start talking randomly. So I made some actual things to help me along the way. So what's going on here is if you have the anhydroretinol in the PKCδ and you have that pathological activation, you're not inactivating it, then the PDHC stays on and you push pyruvate very fast to acetyl-CoA, fast through the TCA cycle. And then there's a buildup of NADH. Okay, that goes up. And then there's an excessive amount of electrons. And you can see there's the coenzyme Q10. So what happens is that anhydroretinol's double bond configuration, like I talked about, it leads to PKCδ staying active. And this inhibits pyruvate dehydrogenase kinase 2. And that's right here in the picture. And PDK2 usually removes a phosphate group from thymine, pyrophosphate on this E1. It's like a part of the complex, and it makes PDHC inactive. But you can see that if anhydroretinol is in there, it inhibits the PDK2, and so that makes PDHC stay active, stay on, okay? All right. And so excessive activation of PDHC drives energy metabolism forward to the point that the electron flow exceeds the capacity of the electron transport chain. So the cell tries to make up for the increased electron flow by mopping it up with coenzyme Q10. And I'm telling you, like, some of the people that allow me to see their labs, I love you guys so much, but they'll be like, my coenzyme Q10 is always so low. And I'm like, oh my gosh. When I went through these slides, I was like, this explains so much. So you're mopping up the excess electrons with coenzyme Q10. So when the electron, I already said that. So when the electron flow exceeds the capacity of coenzyme Q10 and builds up in the intermembrane space, it leaks into the mitochondrial matrix and it forms superoxide. So superoxide is a free radical. And what superoxide does is it's a good thing, should be a good thing. When superoxide goes up, it turns off alpha-ketoglutarate dehydrogenase, which is an enzyme that's in the TCA Krebs cycle. And this is an attempt to slow down oxidative phosphorylation. So it's like a secondary mechanism because superoxide is not a good thing to have in excess. And it's coming from the electrons. So we want to slow down the TCA cycle, which will slow down the production of NADH, which will slow down the amount of electrons flowing through the inner membrane space. So this process, whenever you inhibit alpha-ketoglutarate dehydrogenase, it activates the GABA shunt. So here's the GABA shunt. GABA shunt is where alpha-ketoglutarate will leave the mitochondrial matrix and will be converted to glutamate in the cytosol. And the actions of making glutamate from alpha-ketoglutarate, the enzymes that do this, it's AST and ALT. Those are our liver enzymes, right? So a lot of times in vitamin A toxicity, you see increases in liver enzymes. These enzymes are B6 dependent. There is a non-B6 dependent enzyme called glutamate dehydrogenase. Yeah. And it requires NAD. But the cells are generally, they're in a high NADH state by now. And so most likely we're just going to be using our B6 dependent enzymes. Okay, so once you get to glutamate, then glutamate decarboxylase, which is B6 dependent, will convert glutamate to GABA. And then the backup for that is ALDH1A1. And ALDH1A1 is also an enzyme that metabolizes retinol aldehyde. And so it could be that this enzyme is not available in hypervitaminosis A or vitamin A toxicity because there's so much vitamin A in the body that, it's busy metabolizing that. So again, B6 dependent, we make GABA, and then GABA can go back in. It actually acts as like a signal. So what GABA does is it increases the levels of antioxidant enzymes in the cell. So GABA has been implicated as an antioxidant in the liver, okay? And then GABA is also a neurotransmitter, like a calming neurotransmitter in the brain. So many of my clients struggle with seizure disorder or mood disorders. And I think that it's because of this pathway. And I'll, as time goes on, they have issues with making GABA. So GABA increases superoxide dismutase, catalase, glutathione peroxidase, and the BCL2. BCL2 is something that can stop apoptosis. And then GABA decreases inos expression. So inos is an enzyme that makes nitric oxide. And nitric oxide is kind of like a Goldilocks nutrient, in my opinion. We need some. It's a cell signaling molecule. We need some. We can't have too much. Okay, so when that happens, this is what changes in the cell. So first we'll look at, let's say GABA goes up. So we look at what it does with superoxide. So it will increase the amount of superoxide dismutase, which in the mitochondria is manganese dependent. And then that will lead to the production of hydrogen peroxide. And then hydrogen peroxide is metabolized to water and oxygen. Okay. And then if there were any excess amounts of hydrogen peroxide, then there's also glutathione peroxidase 2 and 1. So we can actually get rid of lipid peroxidations. And glutathione peroxidase 2 is selenium dependent. And they both glutathione peroxidases are selenium dependent. And so that would actually take care of all of the problems, right? And then we recycle our glutathione. We recycle it with NADPH. Okay. So just to sum up that slide, the GABA's antioxidant effects results in the resolution of the superoxide and the hydrogen peroxide by increasing superoxide dismutase, catalase, and glutathione peroxidase activity. So everything will be good if we make GABA, okay? All right. So is it all good though? If you have anhydroretinol pathologically turning on PDHC, is it still good? If you're constantly pushing GABA shunt, that can deplete the cofactor B6, which can lead to a deficiency. It could also lead to low levels of GABA and the inability to downregulate inos. And the nitric oxide from inos can cause a functional B6 deficiency. So this is in a video that I made several months ago after I discovered that my own kiddo had a functional B6 deficiency. So whenever nitric oxide levels go up in a cell, it inhibits an enzyme called methyltransferase or methionine synthase, and it's a B12 and 5-methylfolate dependent enzyme. So you could also have a block in the ability to use methionine synthase if you have B12 or a folate deficiency. Some people have MTHFR C677T polymorphism. They're really bad at making 5-methylfolate. And so if you're compound homozygous and you have both alleles with that particular polymorphism, then you're at risk for struggling in this pathway. So it'd be similar to having like a nitric oxide block. You wouldn't be using this enzyme to go from homocysteine to methionine. Okay, so what happens if that's happening? And by the way, this backup pathway that I'm about to describe that uses betaine, and it's betaine homocysteine methyltransferase, is the enzyme that helps you to go from homocysteine to methionine using betaine. It is active only in the liver, in the kidneys, and then some parts of the brain, just because they've noticed that this is happening in models of MS. So the rest of our cells require that we have B12 and 5-methylfolate. That's an important fact. So if we need betaine, because we have a block from nitric oxide, there's other things that can block this too, like hydrogen peroxide will block methionine synthase. So we'll end up needing more betaine, depending on what's going on in the cell. And we if we don't eat it, and it's in like bran and quinoa and beets. In my own my own family, we don't eat those things. We have like a beet allergy and quinoa allergy and wheat allergy. A kid who's very allergic, and so our whole family is betaine deficient. We have been for 20 years. So if you don't eat it, you make it. And what do you make it from? Well, first you have to harvest phosphatidylethanolamine from your membranes. That's if you're not consuming a source of phosphatidylcholine, because that's where you would get your phosphatidylcholine from. But if you're eating phosphatidylcholine, then you'll convert phosphatidylcholine into betaine aldehyde. And then this enzyme, ALDH7A1, which is NAD dependent, this enzyme would convert betaine aldehyde into betaine. Okay, and so if this enzyme is busy here, and then also, we'll see as time goes on, the cell gets in a lot of oxidative stress. This enzyme also helps to get rid of oxidative byproducts of reactive oxygen species, and alcohol, and hydrogen peroxide. They affect your PUFAs that are in your cell membranes. And so ALDH7A1 can also help you with those. So while you're busy taking care of your lipid membranes and making enough betaine to run very important conversion of homocysteine to methionine, then you end up neglecting your lysine metabolism. And over here, lysine is metabolized to, there's two pathways here, but you can see that they kind of influx into each other. And if you are busy with your ALDH7A1 elsewhere, then you're going to end up having a buildup of this AASA, and that will go to P6C. And P6C actually binds to the active form of B6, P5P, and makes it inactive. And so you end up with a functional B6 deficiency when you have high levels of nitric oxide. So having GABA on board to downregulate INOS would be helpful. Also, I think that people who have high serum A levels, because as your retinol binding protein 4 goes up, you actually cause more nitric oxide in your body too, because RBP4 induces INOS, basically. So in a bad situation, B6 could be a big issue. So here's some other inhibitors: methionine synthase, hydrogen peroxide, nitric oxide, reactive oxygen species, nitrous oxide. And Dr. Ben Lynch is an expert in methylation, so you could check out his websites to learn more about this. Nitrous oxide is what people use at the dentist. It's also used as a bridge in anesthesia, so you can actually ask to not have that bridge. Don't ask, say, I don't want nitrous oxide to help me get sleepy. You can ask for like Versed or something like that. Okay. So let's say that there's some kind of functional B6 deficiency going on, or a depletion of B6, or maybe you're on a medication that depletes B6 from like maybe birth control or seizure medications. So that type of medication could deplete B6. And so you don't finish this cycle. You don't go from glutamate to GABA. There's no GABA anymore in the cell. So ALDH1A1 could be tied up in retinol aldehyde metabolism, and you don't have your backup enzyme either. That's a possibility. So with GABA production decreased, then SOD isn't upregulated, and so superoxide will inhibit catalase. And then the hydrogen peroxide will increase in the cell, and it inhibits aconitase, which is another enzyme in the TCA cycle that converts citrate into isocitrate. And then that causes increased levels of citrate. And then citrate will be exported from the mitochondria to the cytosol, resulting in increased lipid production. So many people have high lipid levels, but that's not the only part of this. So in the cascading events video, I'll talk a bit more about this, but just the methylfolate trap that can happen. Let me go back just a bit to this slide. So whenever you inhibit nitric oxide, if you have 5-methylfolate and it gets trapped inside of, we call it the methylfolate trap, that triggers a whole cascade that induces hyperlipidemia. And so that'll be in the cascade video. Okay. So we got some increase in fatty acids. We're making some more, let's see, we would say like diglycerides, right? Or actually just free fatty acids in the cytosol. Okay, and so here's where I was saying it is a methylfolate trap, and that inhibits GNMT, and that upregulates an enzyme called PEMT, and PEMT uses phosphatidylethylalamine, and that results in lipid breakdowns, like lipid membrane breakdowns, and that'll be in part 3. So this plus lipid peroxidation so we start to have lipid peroxides. It leads to, all these things will lead to leaky cell membranes. And so that's when you will start to see AST and ALT go up in the blood, is when there's problems with the lipid membranes. And also, if your B6 is low, it's potential that there's a compensation going on where so enzymes typically will still run at a very, very, very slow rate without cofactors. Or it might be that the cell senses that you need this enzyme to work properly, and so it upregulates the production of them. So maybe there's an increased production as well as a leaking membrane thing going on, and that's when you start to see AST and ALT go up. And one thing over many years, and I know this is controversial in this vitamin A field that's out there, but the thing that has helped before I even knew about vitamin A, anhydroretinol, the thing that helped my clients the most when they started to have liver enzyme issues was to tell them to eat eggs. I'm not saying that's the only way to restore lipid membranes. You could take a phosphatidylcholine supplement, but eggs have many benefits, including biotin, which is involved in vitamin A in the straw 6 pore. Biotin makes up like part of the straw 6. It helps you to control whether vitamin A is in or out of the cell. So it's a good things about eggs if you can tolerate them. So we need those phospholipids to restore cell membranes. So here's something that I thought. I don't think that a low serum retinol means that you're safe. Safe, I mean from a functional B6 deficiency, because even if RBP4 isn't high enough to induce INOS, there can still be a cascade of events happening in the cell where hydrogen peroxide increases, and that also inhibits methionine synthase. So you could still end up with a functional B6 deficiency, and then the same problem that we'll see in part 3, where you end up with hyperlipidemia. Not everyone gets hyperlipidemia, and that's because some of us are just keeping the fat in our liver. So that's fun. Okay. So the next thing that happens is, while all this is going on, the excess superoxide inhibits catalase, right? And it leads to high levels of hydrogen peroxide, and the excess superoxide becomes a hydroperoxyl radical, so this guy, and then that diffuses into the cytosol, and it's hopefully converted to hydrogen peroxide by SOD1, which is copper and also zinc dependent. And so there's some studies, and this is something I'd have to go more into, but that it's not just that, it's more about the balance between copper and zinc to make SOD run correctly. So that's important to know too. Okay. So if there is a B6 deficiency, then the lack of GABA results in poor upregulation of the antioxidant enzymes. And so this system becomes overburdened. High superoxide levels inhibit catalase, and GABA isn't available to increase catalase levels. And so then what you see is buildup of reactive oxygen species, especially if you don't have upregulation of the enzymes, but also whatever you had in your body that was helping these particular enzymes, even if they are upregulated, you will end up with a depletion of glutathione, selenium, riboflavin, manganese, copper, and zinc too. Don't list it there, but you could end up with depletions of all of these cofactors. And you can see you won't have any of these particular enzymes upregulated if you don't have GABA. So this is interesting. If all of this happens, and you have high levels of hydrogen peroxide and you have iron available, then you're going to make a hydroxyl radical, and that is very toxic to a cell. And during inflammation, the hormone hepcidin is upregulated and it inhibits the uptake of iron. And so it also inhibits the movement of iron through your body. So you either will have low ferritin or potentially high ferritin, and then the ferritin's just like locked into, or the iron's locked into place on ferritin because hepcidin closes iron doors. And so I think this is a protective mechanism for the cell to stop iron from being available, because if iron is available when there's a lot of hydrogen peroxide, then you make hydroxyl radical. So now I got to deal with that guy, right? So how do we deal with that? Well, vitamin E, that's how we deal with it. So vitamin E can rescue us from a hydroxyl radical. So the vitamin E levels, most people start to get burdened with that. I would say the number one symptom of having burdened vitamin E, and it would be like rough, patchy skin. Yeah, also worsening vision, because hydroxyl radicals aren't good for any cells, including the eye cells. Raise my hand, my vision got really bad this past year. So low intakes of vitamin E will lead to cell death. So we need vitamin E, and what vitamin E does is it will mop up the hydroxyl radical and will produce water. So vitamin E can also resolve lipid membrane peroxides or peroxidation. And when vitamin E is depleted, the last defense is gone and the membranes are broken down, and then cytochrome C will be, it itself becomes a lipid peroxidase when hydrogen peroxide levels become excessive. And then there's destabilization of the lipid membrane, and that causes the cytochrome C to fall out of the membrane, and this activates caspase, which is the signaling pathway for cell death. And at this state, excess nitric oxide appears to save the day and actually inhibits the caspase pathway. So again, a Goldilocks nutrient. We don't always want to mop up excessive amounts of nitric oxide. It could be that the nitric oxide is saving us in that moment of time, so it's a bit complicated. There's a nutrient balance. Like I actually use B12 to help mop up nitric oxide, but if I happen to see that on my labs my liver enzymes were high, I wouldn't go insane with the B12. Like I would try to, not go super high, way above the RDA. I can't really give any recommendations because, this is all new to me. So, right. So the cells will start to make new mitochondria to replace the ones that are dying, but these also might have the pathological PKCδ anhydroretinol complex. And so one thing I thought of is that making new mitochondria or sending new PKCδ to the membranes to fix ongoing problems with pyruvate dehydrogenase complex, that depletes PKCδ pools. And I have to study this more, but this is just something I've been thinking of, is that mast cell degranulation, there's studies showing that PKCδ stops mast cell degranulation. So mast cell disorder is a thing that I have in my family. Like one of my kids, he has cold urticaria. That's mast cell degranulation. I'm sure of it. And so I think that his PKCδ pools are being depleted because he's constantly trying to make new mitochondria. So that's an issue, right? We need these particular signaling molecules for other roles in the body, not just to make new mitochondria with new PKCδ in it. There's a pool of this stuff, and if you deplete a pool of a PKC delta, then there's going to have some ongoing side effects. Okay, so this is just me talking a bit about the fact that whenever we have, we should be having like a feedback mechanism, right? And so we should be able to start taking glucose and pushing it through the pentose phosphate shunt pathway. And that should help us to be able to regenerate NADPH, which is needed to regenerate glutathione from its oxidized form to its reduced form. And so it's something that's super important to have the active pentose phosphate shunt pathway. And one thing that restores this pathway is thiamine. But thiamine also will, like if you increase your NADPH, you could also increase NADPH dependent NOx, nitric oxide synthase, and that leads to histamine reactions as well. So, if there's some bad reactions from thiamine. I did dose thiamine for a while. I'm not saying that I'm against that at all, but if you have a bad reaction from it, it could potentially be because you're stuck in the anhydroretinol pathway. And so, you're kind of like flimsy maybe on PKC delta, and then you add in bivlotimine, and then, it just pushes your histamine load. And then I also thought it was important for this video to go ahead and consider the fact that there's an interplay between our pentose phosphate shunt pathway making NADPH here and our glutathione recycling and our vitamin C and our vitamin E. There's an interplay there. And so it's about balance. So for me in general, I know that my daughter who has a genetic syndrome, she's really bad at making glutathione. So if I give her a large amount of vitamin C, then you can see that here's glutathione in its reduced form, right? If I give her a lot of vitamin C, then I'm going to use up her glutathione going from ascorbate to dehydroascorbate. So there is an interplay. You really shouldn't go super high or, on one nutrient. Same thing with vitamin E. Like there's studies showing that vitamin E is an antioxidant, and then at large doses, it becomes a pro-oxidant. And that makes sense if you think about this interrelationship. So here's alpha tocopherol, right? But if you have an imbalance of vitamin C, then you can see that you're going to have an imbalance in your healthy reactivated form of vitamin E. So that's something to think about. Okay. So the next video is going to be part 3, the downstream effects of anhydroretinol. Just to sum up, I think, and this is in a previous video, but I'll just kind of sum it up here. I think that the things that are going to help pretty immediately to resolve, mitigate would be prevent, but to resolve the reactive oxygen species actually would be vitamin E and vitamin C. Hopefully, you're going to be making some glutathione. That's a whole nother thing because you have to have B6 to do that. But I think that vitamin E and slow C could be helpful. And then also coenzyme Q10. Those are the 3 things that I think would be helpful to stop some of the damage that's occurring. Okay, so I'm going to try to record all these in one day. So I will see you in the next video as soon as I can pull up. I don't know why I can't pull it up. I think there it is. So I'll see you in the next video. I hope this one was helpful.